Afternoon, and welcome to the lightning round of genetics review for animal lovers. My name is Hannah, and I'll be your animal loving guide through a quick look into the Lyon Hypothesis, X-inactivation, and X-linked inheritance. Let's get started. So first, I'm going to talk about the Lyon hypothesis and X-inactivation. Mary Lyon created this hypothesis with the help from an observation by Murray Barr that each cell with two bodies seemed to have something that looked very dark, stained, and condensed towards the outside of the nucleus that he referred to as a body. The inactivated one would become known as the Barr body. This is a widely accepted rule for placental mammals today in the field of genetics called the Lyon hypothesis. And it basically just means that when two X's are present in a female cell, one is turned off like a light bulb or inactivated so that there's only one instance of gene expression. Because otherwise, with females having two X chromosomes, there would be double the amount of gene expression going on. X-inactivation is random. This is a super important keynote, and that means that no female animal will have exactly the same traits expressed because the X that is inactivated in each cell is different, and the traits that are expressed are unique. Tortoiseshell cats are unique, beautiful creatures that have a mixture of mottled black and bright orange-yellow fur. This is because of X inactivation occurring as the tortoiseshell color is linked to the X chromosome. The tortoiseshell coloring is known as an X-linked trait, which means that the X chromosome is responsible for passing on and showing the trait, and thereby only allowing girls to be tortoiseshell colored. Having the expression of only two dominant alleles for the trait would yield a black coloration, while two recessive alleles would yield a yellow coloration. But the mixture of having both a dominant big B allele and a recessive little b allele on the two X chromosomes passed on will render the beautiful coat coloring that we know as tortie or tortoiseshell. With only one X chromosome being activated, the two alleles are going to be expressed randomly based on the X that is being expressed or not turned off. Multiple cells randomly express one of the two alleles for black and yellow fur based on the X chromosome that is inactivated. I'm going to show you guys an example of how tortoiseshell kittens, which remember are only female because they have to have both X chromosomes for those alleles to show up, are never exactly the same, with help from an autosomal trait of coat length that when dominant shows up as long hair and when recessive shows up as short hair. So this is a branch diagram at Punnett Square, but it shows that a cross between a yellow short-haired female and a black heterozygous long-haired male would end up yielding 1/2 or 50% chance of each color, and a 50% chance within that of each length of hair would end up with 50% female kittens, both of which were tortoiseshell colored, and 50% yellow kittens, both of which were male. It's more specifically 25% tortoiseshell females with long hair, 25% would be tortoiseshell females with short hair, 25% would be yellow long-haired males, and 25% would be yellow short-haired males. So the question, again, why aren't these cats the same pattern? Because each kitten has many different cells with which each present their traits differently, and the amount of yellow alleles and black alleles being phenotypically portrayed is different. All the patches of yellow fur and black fur are made individually by lots of light bulbs or cells with X's turning off. Thank you so much for listening to this video, and give it a like and subscribe. Thanks!